Good afternoon, everybody. My name is Abby Bronson, and I'm the Senior Vice President of Research Strategy at PPMD, and I'm delighted to be able to introduce this afternoon's webinar on microdystrophin gene therapy for Duchenne muscular dystrophy. Before I introduce our speakers, I want to let you know, I think there's a little bit of housekeeping to do. We will have time for questions at the end of the talk. So if you put your questions in the chat box, we will take as many as we can at the end of the webinar. We also will answer all those that we don't get to during this webinar by email after the webinar is done. And when we upload the recorded webinar, we'll also have a Q&A that goes along with it. So now it's my pleasure to introduce our two speakers for this afternoon. We have Dr. Jerry Mendell, who is an attending neurologist at Nationwide Children's Hospital. He's the PI in the Center for Gene Therapy at the Research Institute at Nationwide Children's. He's the director of the Paul D. Wellstone Muscular Dystrophy Cooperative Research Center, co-director of the Muscular Dystrophy Association Clinic, and professor of pediatric neurology, pathology, and physiology and cell biology at the Ohio State University College of Medicine. Dr. Mendel was the first person to study viral-mediated gene therapy for muscular dystrophy in humans, and I think many of us have read some of the papers that came out of some of that work earlier. He's made fundamental contributions to clinical research in molecular genetics of neuromuscular disease. Dr. Mendel is named among the best doctors in America. We also have his co-PI, Dr. Louise Rodino-Claypack. She is an associate professor of pediatrics and molecular biology at Nationwide Children's, and I think she probably is the person that really keeps Dr. Mendel on his toes. So I'm really happy that both of them can be here, and now I'll turn it over to our speakers.
Well, Abby, this is Jerry Mendel. It's a pleasure to be here, and I think we have an exciting story to tell. And you're right, Louise Rodino-Claypack has been really a principal mover for this clinical trial and done much, if not all, in her lab on the preclinical. And what I'll try to do during this talk is to, one, address questions that might come up during the trial just from previous experience of presenting gene therapy studies, and also tell a story about how you get from the clinic, from the lab to the clinic. I think that's an important piece of information, especially for patients and their parents who are interested in moving ahead. So that's a picture, and it does tell a genuine story that we have clear plans on how to conquer Duchenne dystrophy. I have been working on this not naively to say it's been 50 years. I was at NIH in 1969, saw my first Duchenne patient there, and from that point forward, I was committed to making a difference for this disease. Louise is in the middle there on that picture, and she has been working side by side with me for the past 15 years, and really made an important difference. I wanted to put this slide up because these are questions that I hope we can address during this trial discussion. And if I haven't answered all these questions, I'll be happy to address them afterwards, either by questions that you submit or by questions that Abby raises. So a common question is, where do we get the gene? And I'm going to show you how we get it. How does it get to muscle? That's a very important question. Is gene therapy safe? A critical question that patients often want to know. And then another issue that frequently comes up is, how many times do we have to give the gene? Is it one time, multiple times? And I'll try to address these questions. So let's tell the story. The story begins with the virus that we use. And there are three common viruses that are used for clinical delivery of genes. Adenovirus was at one time the most common of all the viruses that was used. That's the top picture there. It had advantages because, as I'll get to the bottom one, you'll see that we have limitations for the bottom one. That has to do with how much the virus can actually carry in a gene therapy trial. Adenovirus is wonderful because it can carry any size muscular dystrophy gene. It has its downside because it is an active infection when it's entered into the human body, and that resulted in a major disaster back in 1969. And since that time in 1969, it's virtually never used for what we call a monogenic illness, an illness caused by one gene in the neuromuscular field. It's mostly used to treat cancer now. The second one is called lentivirus. Lentivirus also has a large packaging capacity. It has one specific downside, and that is that it integrates into the human genome. What that means is that when we put that virus in there, it will actually deliver its gene to the chromosome, and in that site in the chromosome, it's very safe in there for the gene. But the gene, as it integrates into the into the genome, can actually disrupt other genes, and that is the reason it's not used very often. Adeno-associated virus has been the principal delivery tool that we've used for gene therapy over the past, actually, our first gene therapy trial was in 1999, and since then, adenovirus is really the key player for moving genes, moving muscle and nerve genes. It has the downside I mentioned. It's a small packaging capacity, and you'll see that how that impacts us. This just elaborates a little bit more on the gene. It's small. This packaging capacity is 5 kilobases. Now, it probably doesn't have a lot of meaning for most of the listeners on this webinar, but what it means in terms of practical delivery terms is that the Duchenne gene, the DMD gene, is over 11 kb, and so we can't deliver the entire gene into the through adeno-associated virus. We'll come to how we deal with that in a minute. It is non-integrating. That's what I said was the downside of lentivirus. But when it gets in the cell, for all reasons that we know of now, it persists in the cell as what's called an episome, and you'll see a picture of that in a minute. But it doesn't integrate, and it can't disrupt other genes. So in that sense, it's incredibly safe. There are over 100 serotypes of AAV. What a serotype is, we take the virus, and if we just change the content of the capsid, which is the surrounding part shown up there on the right-hand side of your picture, with little spikes coming out so it can deliver well. In those spikes, or in that shell, if we just change the amino acids by 3 or 4, we change the serotype of the virus, and that's potentially important, as we'll come to learn in this discussion. If there is any loss of gene expression over time with the virus that we use. The common serotypes that we use are AAV1, AAV5, 6, 8, 9, and RH74, and they're written down there. They all have a slightly different purpose. AAV9, for example, is excellent for delivery into the nervous system. AAV-RH74, the one we tend to use, it's important here at Nationwide Children's Hospital because AAV-RH74 was actually isolated here from a rhesus macaque monkey, and that's where it gets the name RH. And when we use that in a clinical trial, there are fewer people who object specifically to using that virus because we basically claim ownership to it. So there's less conflict when we have a trial and potentially have success from a trial. So that's the reason we use these different ones and RH74 in particular. Now, I mentioned that we have a limited packaging capacity for the dystrophin gene, or the DMD gene, as it's officially called. And here is the situation. We call it microdystrophin because we have to take and trim it down. And if you see that very bottom picture there, that's the trimmed down version of that. And if you look down here, what you see here are certain numbers. You see 1, 2, 3, and then separated by this number 2, and then number 24. And if you look up here, here's the gene. And what it shows is there's 1, 2, 3 there, and skip all the way over here to 24. So the gene is actually spliced down, and we splice out all of this middle region here. And what we found, and you'll see in our treatment in the laboratory, which Louise has run pretty much exclusively, that this actually expresses very well and corrects the defect in DMD very well. So there are advantages. Now, what is the disadvantage? It's not so much how much we can correct. It's in the initial trial, we have to exclude patients who have any mutations or deletions in this part of the gene or in this part of the gene. That's the part that is microdystrophin. But here, anybody who has a mutation in this very large part of the gene is eligible. Because if we express the gene, this microdystrophin, into this part of the gene, then the body does not produce any antibody to what we express in here. And so there are advantages. If the gene does not specifically express in here, so it's only expressing here, and so there's no conflict in any part of the gene that that now is expressing. And if I'm explaining that well enough, if there were a mutation here, go ahead.
Is it kind of like when you put this microdystrophin in, you don't want some part being created that the body has never seen before, right? Is that right? Is that a way to think about it?
Absolutely correct.
Okay, so if you had a mutation like in exon 3, then, your body's never seen beyond 3. So then if you put in the microdystrophin that you make that goes all the way up to exon 18, there's all that new stuff from exon 3 to exon 18 that your own body has never seen. That's a very simplistic way to think about it, but maybe it's a way to understand it.
Well, it certainly clarifies what I was saying, and I appreciate that.
And also, can I ask one other question? Is that when you say between exons 18 and 58, does that include mutations within exon 18 and within 58? Or, where's the actual border?
Anything below 18, so that's 17 and 59. Now, the reason that's so important for patients is most of the mutations in Duchenne dystrophy occur within this range of 18 to 58. So it's clear that we will catch the majority of patients with the disease. But extending that one step further, the truth is that we might be able to extend this out for some patients because mutations can often be leaky. So by that, what I mean by leaky is that even if there is a mutation down here in exon 3, for example, the gene there might be a slight splicing defect where we do get protein down here, so that when we put our gene in, it's really not, the body has seen that before. We won't entirely know that until after our first trial is over, because what we intend to do is we want to frame the first trial for the maximum benefit of success. If we can show success here, then we can start treating patients outside this region. And my own opinion is, quite frankly, that we would have a good chance of treating patients outside these specific regions. So I think we can do it, but we want to frame the first trial for the maximum rate of success. Now, also notice that another important part of this is called a cassette or a construct. This is what we fit in the virus, and I'll show you how we do that on the next slide. I didn't want to show the next slide yet, but what we have here is a promoter. When we put the gene in, it must have a promoter in order to express. This is called an MHCK promoter, and what we know about the MHCK promoter is that it enables expression not only in skeletal muscle, but in the heart. And by expressing in the heart, in Duchenne dystrophy, that's extremely important because we have to correct the cardiac muscle. Many people thought one of the downsides of gene therapy for muscular dystrophy, for Duchenne dystrophy, was if we corrected skeletal muscle and didn't correct the heart, we would be putting too much stress on the heart. So the fact that we can correct both at the same time is a major, major advantage for patients. Now, so I showed you the virus. I showed you the gene as we whittle it down to what we call microdystrophin. And now we get a little bit more detail on actually how we make the gene. This is a common question. Where does the gene come from? So if we start out with a virus up here, this is the AAV capsid that we talked about. And here you see it up here. And inside this, the AAV only has two of its own genes. So what we have to do is to get rid of the viral genes. We don't want the virus to work when we put it in. Not on its own. We don't want rep means replication, cap means making the capsid, and if we put that in, we'd make more of these guys. We don't want more of those guys. We want more of what we'll see, I hope, on the next, hold on a minute, I think that works, yeah. We want to remove rep and cap, the viral genes, and we want to replace the viral genes with microdystrophin. Now we've got what we want. We're going to take this microdystrophin now, we're going to put it back in that capsid or capsule, and then we're going to have a complete recombinant AAV that contains the microdystrophin and the viral capsid. And the way we do that now, I'll show you on the next step, is that we take the microdystrophin gene now and we put it in what's called a plasmid. A plasmid is kind of a hard concept to understand, but it's basically a circular DNA. And we actually put the viral capsid, we put the microdystrophin gene into the plasmid, and then we take it to our vector manufacturing facility here. And we have a very large vector manufacturing facility at Nationwide Children's Hospital. We started out with a small one, and now in the last 2 years, we've built it into a very large one. And those arrows show, dadgummit, that shouldn't be on there. There it is. What we have now are multiple production rooms within the vector manufacturing facility, and we can actually make 4 different runs of gene at the same time. So this gives us a very large capacity. We also have a cell-based therapy over here. So at some day in the future, we envision taking the virus or taking the microdystrophin and perhaps putting it in a cell so that we can do cell-based therapy. That is in the future, but we're prepared to move that ahead. We also have rooms in the viral manufacturing facility where we clean up the gene, and when we take it from the plasmid, bring it to here, load it up into the capsid, and the way we do that is shown on the following slide. We use a manufacturing production that is called triple transfection, and we take 3 different plasmids, one that holds the microdystrophin gene, one that holds rep and cap, and you'll see why we have rep and cap in a separate plasmid, and an adenovirus helper. We need all these 3 components because we put that into a cell line called the 293 cells, the human embryonic kidney cell 293 line. We put these 3 plasmids in here, and what happens is when we put that in with our plasmid that contains the gene, what comes out are the capsid that we have from actually these rep and cap genes, and the gene that we put in the plasmid, and here's our microdystrophin gene now within the viral capsid. Now, there's several important things about this. When we put this in, this is what we deliver to patients, and in this delivery system, each one of these capsid that contains the gene is really the number that you'll see in a minute, which is in the millions now when we deliver this to the patient, and really determines basically the success of our gene therapy. So how does that work? Well, our vector manufacturing facility then goes to Louise's lab and says, will this work in mice, especially in either normal or experimental mice? The experimental mice for muscular dystrophy are called MDX mice, and it works like this. So here's the recombinant AAV containing the microdystrophin, and that's what it looks like. It's injected usually into the tail vein of the mouse. It goes to muscle, and in muscle, the muscle then delivers it to the nucleus. The capsid now is degraded, and the gene, which is the microdystrophin gene now, is delivered to the nucleus. Now, without getting too complicated, this has to be further transformed into what I mentioned before was the episomal formulation of the gene, and it has to be double-stranded just like your regular DNA is. And from this site now, and we hope forevermore, this double-stranded DNA then acting just like any other gene in your body then will make the microdystrophin. If it does that successfully, then we have a winning combination for gene therapy. So you can see where the virus comes from, how it's made, and how it's delivered now. In patients, we deliver it exactly the same way, and this is exactly what we hope to see in patients. Now, for the next step, what we have to do is we have to make sure this gene is working in mice. If it is working in mice, then we can go to the FDA and say we have a combination where the recombinant AAV carrying the microdystrophin gene will work in mice, and this is an example of a sequence of pictures that shows that it is working. This is an untreated MDX mouse, and you can see across that either in muscle or heart or the diaphragm, which are really the critical parts of the muscle that we want to express in to get the maximum effect for the patient. So the MDX mouse doesn't show any stained muscle fibers in there. Here's normal, what it looks like. These round objects are muscle fibers here, each with dystrophin around each muscle fiber. And in the heart, which has basically smaller muscle fibers in the heart, we see expression in every muscle fiber in the heart and then in the diaphragm. Now, we want to see that same picture when we inject it into the MDX mouse. And sure enough, when Louise did that, she saw very good, robust gene expression in skeletal muscle, heart, and diaphragm. And then this bottom picture here shows that that dystrophin will link with other proteins in the muscle, and that's very important because if it links with other proteins in the muscle, it's more likely to rescue the muscle fiber so that there is no concern about muscle degeneration at a later time. And to further validate that, the FDA wants to see how much are you going to inject in the patient? Can you inject more if you need to? If that doesn't work, and what this slide shows is that the dose we plan to use in the clinic is working very well in the heart, in the diaphragm, in the limb muscle, couple of different limb muscles, and we can get both more effective by raising the dose and safe gene expression by using even a higher dose. This is important because potentially, and you'll see the way we plan this clinical trial, we'll do biopsies early and late in the clinical trial. And if we don't have very good gene expression, or we think we should have more, then we can raise the dose. Now, the other thing about this is, how do we know this is safe? And during the testing of the gene to get to clinical trial and to show to FDA, we take every mouse that is injected with the gene, and we do a complete and full autopsy on every one of these mice. This is a tremendous amount of work, but we can take the safety data now and go to the FDA and say that, do we have any complications from the injection? Do we have any inflammation? Do we have any downside? Do we have any off-target effects? And in preparation for the clinical trial we're about to start, when we presented all this, and we present a very large package to the FDA, we had no off-target effects. The other thing that we want to show, and the FDA is very big on this, the FDA wants to make sure that we can return the muscle to normal in terms of function. So we look at the ability of the muscle to contract in comparison to normal muscle, and we look at the force generation of the muscle. What this slide shows basically is a dose escalation, and at the highest dosing level, we can reproduce exactly the same amount of specific force that we have in normals, and this is the diaphragm, which is really protective for the patient, and this is a limb muscle then where we get, in the limb muscle, we get very close to complete return of force generation. So all this information is then taken to the FDA and taken in the form of what we call an investigational new drug application. They look over all the safety data, and from that point, we plan the clinical trial. And we're really right at this point now where we're getting the final okay from the FDA. Now, a trial like this, the first in human trial for a microdystrophin using the MHCK7 promoter, this is what we call a phase 1-2 trial. Phase 1 means it's safety. Phase 2 means really asks, does it work? And that's what we'll be asking in the clinical trial. This is the protocol synopsis for the clinical trial, and it will be a phase 1-2A clinical trial. 2A means that there won't specifically be a placebo group for this clinical trial. And, of course, from a patient point of view, that's a major advantage. So every patient in this trial will be treated by AAV-RH74, MHCK7 microdystrophin. We'll put it into a vein in the arm. This is actually the nice part of this trial. This is very simple administration or perfusion of the patient. We take a regular IV, put the regular IV into a vein of the arm, and we can inject it very easily. And
Dr. Mendel, is there, I thought you were going to the next slide, but I'll quickly ask my question. Is there, when you say the sample size, are there placebo patients in there or no? They're all treated patients?
There's no placebo control group in this clinical trial, and that's why it's called a phase 1/2A. A phase 1/2B is where there is a placebo group. And in many ways, this is much easier on patients, also easier on us, actually. But what we have here, we designed this trial so we could ask a question about the patients in terms of comparison between very young patients. You'll notice we'll be admitting patients who are 3 months to 3 years, and we'll be admitting patients who are 4 to 7 years of age. And we chose to do this because we think that obviously we learned from other clinical trials that high dose and early time delivery is important in terms of predicting benefit, but we wanted to compare this with the more common age groups where patients are mostly diagnosed at age 4, so we didn't want to exclude those, but we want to be able to compare the very young patients with the more common age of diagnosis where we first see patients at age 4 to 7. So this will be a very powerful trial in comparing the two groups. We also wanted to make sure that we would have access and patients would have access to the clinical trial by using 2 different centers, one here at Nationwide Children's and the other at WashU St. Louis Children's. And I chose to collaborate with the St. Louis Children's. We have a very long-term relationship with the group at WashU at St. Louis Children's. I've worked with that group since the mid-'70s, actually, and so one of the advantages is that all of the studies that are done here with what we think are very rigid, practical safety criteria with good efficacy outcomes, we are confident that those same things will be done by Ann Conley at WashU, and we'll be able to have a very good collaborative relationship, and she's an excellent investigator. And the primary objective for this trial is safety. I mentioned that this will be infusion of the gene over 1 hour. A couple of things about the inclusion criteria. I already mentioned that we'll be including patients only who don't have mutations between 18 and 50. Only mutations between 18 and 58. There'll be some other criteria that patients have to fulfill, and these are really more FDA satisfaction. Most Duchenne patients have CKs that are over 10,000, so CKs over 1,000 is a minimal criteria. Obviously, there'll be males who are enrolled in this trial of any ethnic group, and they must be able to cooperate for any clinical assessment. There'll be a couple
Can I ask a quick question about the 18 to 58 again? What happens if it's a nonsense mutation, like a premature stop codon, in between that, in that region, like at exon 30? What happens? Are they eligible?
Yeah, we don't think there'll be any contraindication to that.
Okay, perfect. Thanks.
And one point about this slide that I just bring out, we'll be pre-testing all patients to make sure that they haven't been had previous exposure to AAV, because we don't want to go into this trial with an antibody in the bloodstream to RH74. We have ways of dealing with that, incidentally, but on the first trial, we want to be able to line that up to have a favorable response. And then since we'll be able to do very young babies in this trial, If the mother has been has had pre-existing exposure to the virus and is carrying an antibody, we know if that's carried through by the breast milk, we'll ask the mom, we've had experience with this, we'll ask the mom not to breastfeed for a period of time until the antibody clears, and then we can retest the potential candidate for this trial. So I mentioned about the vector being infused very simply. We may, for the very young infants, sedate the child for the infusion. We'll work closely with anesthesia here if that's necessary, and we'll give the microdystrophin gene at very high dose IV. Just a couple of things about, the studies tend to be intense in the sense of frequent follow-up, and those are the follow-up days there, 7, 14, 30, 60, 90, 180, and then continued on at 9 months, 12 months, 18, and 24 months. The FDA might ask for longer time periods of follow-up, and we'll add that in if they do. We do a muscle biopsy before we start the trial, when we'll do one biopsy at 12 weeks and one biopsy at 24 weeks on different groups in the patient. Some will have, they'll have one pre and one post. There'll be a certain number that have the 12-week and a certain number that have the 24-week. And I mentioned the reason for doing the 12-week is that we don't have good gene expression. We will increase the dose for patients who enroll after that time period. And that's why I showed you we have data that gives us what we call the clinical dose and another dose at higher levels. We'll be monitoring the patients very closely. We're doing the 100-meter walk and the Bailey 3, depending on the age of the patient. So that's pretty much the picture of what we have. We're very optimistic about the benefit of this trial. We think that all of our preclinical data looks very favorable for producing a clinical response. I mentioned about what a couple things here that often come up are what if we don't, what if after we put the gene in it stops working? And one of the things we've learned is that the problem is when we put the gene in, the patients will develop an antibody to the virus. But we've learned that we have ways of getting around that. One is that we can physically remove the antibody by a method called apheresis or plasmapheresis. And we have a window of time to get the virus in after that. We will be further testing that for efficacy as this trial moves forward. We'll be doing additional studies in non-human primates to make sure we can do that. We have ways of blocking the antibody. And I mentioned also, I showed that slide where there are different serotypes of AAV, and we could switch the serotype from RH74 to a different serotype, and that could be one way around the AAV. The AAV antibody will be directed against the specific serotype. And then the final issue, if there's any other questions, is about what are the other potential methods of treatment. And one of the hottest topics in all of molecular therapy for gene correction is a method called CRISPR. CRISPR works differently than delivery of the virus per se. This actually corrects the gene mutation within the genome itself. So it is a way of actually correcting it, and it corrects it for a more indefinite period of time than viral gene therapy, where we replace the gene per se. But it's still quite a ways off at this point. We'll work very closely now with Eric Olson on designing a clinical trial for CRISPR, but it's still quite a ways off, and I would say we'll have data that is very clear from our gene therapy trial that is well in hand before CRISPR comes to clinical trial. So we'll be able to compare that at some point in the future head-to-head. But we'll know whether gene therapy works before CRISPR is introduced into the clinic. So I tried to anticipate and address most of the questions. I don't know, do you want to add anything, Louise? Or Abby, I'm available to answer any other questions.
Louise, do you have any? Or any comments?
No, I think covered it well.
So I have, so when we start this phase 1, like how soon do you think, you said the trial lasts 2 years, so to, wrap up all the data analysis, and to get the final official results, it would probably be 3 years, right, from the last patient in. Yeah. So probably, depending on how long it takes to get enrolled, it could be anywhere from 3 to 3.5 years. So, but when would you expect to sort of start seeing some hints of when it might be working, if that's a fair question? If it's not a fair question, don't answer it.
No, it is a fair question, and to go back to enrollment, we anticipate that we're asking the FDA if we can enroll patients every 2 weeks.
Okay.
So we should have very well in hand a pretty good cohort of patients within a relatively short time. The second part of that question is when we know if the virus is working. There are 2 pieces of information about that. One, at 12 weeks, we'll do the first biopsy, and at 12 weeks, we could potentially, we'll have what, we'll have the first patient first patient will be able to, that'll be 10 weeks. When will we have the first 3 patients at 12 weeks, Marcus? I thought you were on your calculator there, about 18 weeks. So we'll have a good idea about 18 weeks after we start the trial, whether we're getting good gene expression from the first 3 patients. And then we'll know about whether we should change the dose. We don't want to go 3 years into the trial and then say, I wish we had changed the dose. So we'll know.
Right. And I know you talked a little bit about the beginning about the intent is to expand this so those mutations on the ends weren't excluded. And, right now we're doing this in a pretty young cohort of patients. So, and I think the overall intent is to have gene therapy be available for all types of patients. So how would that play out? Like, you do this trial, when would you start sort of opening up the inclusion criteria? When would you start maybe adding different cohorts of patients? What would that scenario look like?
I think by 12 to 18 months, we will have a good idea about efficacy, and we could easily go back to and whether there's any off-target effects or any significant adverse events, which we really don't think will occur. But we can go to the FDA at that point and open up the trial with an amendment. And I'd be pretty intent to do that because I think that we're trying to line this up so we get favorable results, but I think we can open it up to other patients in a shorter period of time, certainly than 3 years.
And would you start with adding in those mutations that were initially included, or would you add in? That's probably the first opening you do.
I think it'll depend on interaction with the FDA.
Okay.
But I'd be very eager to expand the trial because I think if we did that, we would be we would be able to reach more patients, and we don't want to make this exclusive. That's one of the limitations of CRISPR, actually, is that the first trial will be targeting a single mutation or a single group of mutations that will skip 51. And our trial is more inclusive than that. So we can target more patients, I think, with gene therapy if it's effective.
And if it's effective in this group of patients, do you see it expanding into older boys, non-ambulatory boys, boys that have more severe, more progressed disease?
I think that's a critical question, is whether what we can do in that age group. And I'd certainly be very eager if we can get diaphragm and other muscles that control breathing and heart and good gene expression in the heart. We can, have a major impact on the older boys.
Mm-hmm. So how are we going to study the cardiac function in this trial? We're collecting some cardiac endpoints?
Yes, we're doing cardiac MRI in this trial.
Okay. Let's see. So have you gone into any non-human primates or done any work on dogs? Because I know that's also, those models are even closer to what the human is.
I'll let Louise answer that.
Okay.
We've done extensive non-human primate studies with this particular virus. With various genes, we've done over 100 animals and found no adverse events. We've also done specifically with this construct in primates with good gene expression distribution and very good safety. So we've definitely gone into larger animal models and shown it was safe.
I'm trying to see. What about, we have a question about the blood-brain barrier. Do you anticipate any kind of effect for boys that might be suffering from any cognitive delays? Could gene therapy possibly help them?
That is a highly speculative issue. It's an interesting and important question. If we extrapolate from our SMA success, we get, in SMA, we deliver the gene in exactly, well, let me just not use any abbreviations here for those people who aren't accustomed to what SMA is. There's a disease, the most common severe disease in infancy that is inherited in a different way than Duchenne. Duchenne is the most common severe X-linked or muscular dystrophy, most common severe muscular dystrophy in childhood. In SMA is the most common severe infant form of neurologic disease, where the infants don't live past 2 years of age. And in that trial, what we've done is we've taken AAV with the gene that is missing in spinal muscular atrophy, or SMA. We've delivered it. It goes into the nervous system and will correct the nervous system at every level. And the levels that we can document are corrected are in the brain and all throughout the spinal cord, in the neck region of the spinal cord, in the thoracic region of the spinal cord for breathing, and in the lower lumbar region of the spinal cord. So we know we get very good effects throughout the nervous system. And one might speculate that with the virus that we're using, we might also be able to achieve similar results. Now, that would require a change in promoter in order to make that happen, because the MHCK promoter is a muscle-specific promoter. So the way we have the trial designed right now, it's not going to go into the brain. But that could be changed. And also certainly if we get success, we'll want to explore those issues about changing the promoter and maybe even changing the viral serotype. So, but I think it's a very important question, and one that certainly, if we can achieve that good a success in the heart and diaphragm and limb muscle, I would say, we've opened up the avenues to really accomplish virtually the disease in its entirety.
Yeah. So another question, you talked earlier about the vector going in and it's an episode and it integrates into the cell and it persists for, we don't know exactly how long, but for a good amount of time. So as a question came in about whether muscle cells are division, will there be any kind of dilutive effect on the microdystrophin? Or the effect of gene therapy, or even for that matter, as a boy grows and muscles get bigger, will there be any dilution, a dilutive effect, would you expect, or is that not the right way to think about it?
So one of the ideal things about muscle is that muscle cells in and of themselves do not divide. So when you are transduce the nucleus with this episome, it will stay there. So you won't be like the liver divides and then you'd be losing the episome. In muscle, that doesn't happen. Now you do, over time, as there's damage, incorporate satellite cells. From our studies, we know that a proportion of satellite cells, not a high amount, maybe less than 10%, are transduced by AAV. So some of those satellite cells, these are muscle stem cells in the muscle, will have the virus and some won't. So there is the potential for, over time, to get some dilution of effect. We just don't know. We'll find out over time.
Okay.
So that's a question.
And what about...
that comes to the issue of redosing again, Abby. That I think in the long run you're going to see redosing, and that's one of our major goals as we move forward here, is to study the whole issue of redosing. And would redosing be most likely done with a different serotype or different delivery mechanism, or what does your crystal ball tell you? I think those things are to be explored. We know we could potentially redose with a different serotype, but we can get rid of the antibody and redose, or we can suppress the antibody and redose. There are all those possibilities. And what we'll be doing in the next couple of years, really, as this trial is underway, we'll be exploring those issues. It's a critical question. We get asked it every day, which gets to that one question that I had in the beginning is, how long does it last? And that's basically what you're asking. And at this point, we have data. There's in some experimental studies, the virus has been there and continued to express for as long as 15 years. In clinical trials, in the hemophilia clinical trial, we have viral persistence for 3 years. And in the SMA gene therapy trial, we have viral persistence for over 3 years. So that's a pretty long time. And if it would change after that, I don't think we can say, but we've got pretty good evidence of long-term expression and safety now in other clinical trials.
Okay. I asked about nonsense mutations. What about duplications? Would they be just considered another mutation, and as long as it's within that region,
well, a couple
it
things
could be included?
About duplications that I think are interesting and potential. Duplications tend to be a little bit leakier in the sense there might be some gene expression, and if it's a frame shift mutation and doesn't produce any, it's not below 18 and not above 58. Then the same rules apply. But if if it's a frame shift mutation, I think that in the long run, I don't think for our first clinical trial, but Kelly's looking at me. If it's a frame shift mutation, we can enroll them. But I think there might be intrinsic protection in the duplications with a little leak. If there's a little gene expression, that protects the patient, actually.
Right.
Thank you.
So this is all so exciting, and I know that everyone's really looking forward, to just starting this. When are you projected to start this phase 1-2 study?
submitting an IND.
Don't say that.
Yeah.
And what does it depend on? What are we sort of waiting for?
Well, we're waiting permission from the FDA, basically.
Okay.
And
all right.
We had scheduled this trial to start in late October, early November, and so far I think we feel like at this point we're on target to do that. If the FDA comes back, I think it's important for your audience to understand that there's something called I mentioned about the IND, the investigational new drug application, but in preparation for a clinical trial, there's what we call a pre-IND. A pre-IND means that we submit the entire plan for the clinical trial long before this point in time. At the pre-IND, we discuss all of the potential guidelines that the FDA puts in front of us to follow in order to get to the clinical trial. And as far as we know, we have followed every one of those guidelines and met every one of their criteria that they put in front of us in order to get to the clinical trial. So they'll investigate all the documents that we submitted now, and they'll say that they're either completely fulfilled, or you missed this one, or we want to see this. But most of the time, we haven't had much pushback. We have 7 INDs in the clinical center here. So we're not an inexperienced group, and in general, when we get this far, we anticipate pretty much that we can move forward, and that's kind of where we are. But the FDA's got the final word.
Okay. And so if we think forward and we do this phase 1/2 and it's successful, would there have to be a placebo-controlled trial at some point, or you just don't know yet?
I'd say we don't know. If we have spectacular results, I would say no. If we have equivocal results, I would say yes. And we're planning on very good results. I didn't say spectacular. I'd like to say that, but I'm trying to be cautious. So,
right, exactly.
I believe that we can have very good results.
Yeah, and so and I know that I'm reading the questions, and I still think there's a lot of specific questions coming in about my mutation is here, am I eligible? So I think we're going to have to do a post-webinar really detailed sheet about who's included, types of mutations, nonsense deletions, duplications, and where, because there's still a lot of questions, so there must be some confusion about that. So we can deal with that offline. So everybody who's asked questions about that, please check back, because we'll write up something and post it on our PPMD website. And I think, I guess, the other thing that has come up, I did have one other question. What about, why did you make the decision to go into the 3-month to 3-year-old cohort, that very young patient? Are there advantages to doing that?
Absolutely. I mean, what we learned from our SMA trial that was very successful, where we really truly saved lives and saved function, that the patients in that trial who were the youngest and who received the highest dose were the ones that did the best. So when we planned this clinical trial, we wanted to simulate what we did in the SMA trial. And in the pre-IND, we asked the FDA if we could do 3 months to 3 years. They said yes. And then we wanted to be more inclusive in the trial, so we wanted to add the 4 to 7s for the reason I said. I mean, most of the Duchenne boys are really diagnosed at 4. Unless there's something that brought it to attention. It could be an inadvertent blood test. It could be a family history. There were other things that brought it to attention, or a parent who's asking questions about delayed milestones and you have an astute doctor who does the right testing. A lot of times, as you know, some of the early milestones that are not met are attributed simply to a delay in milestones where a prediction is made that the boys will catch up and never do. Mm-hmm. That's right. I mean, still, if you look across the board, most boys are diagnosed at 4 and above. So, okay. But I think the other thing about our trial, we're very hopeful that it'll change the playing field so much that we'll have newborn screening, in which case it'll outside. I mean, then we really have a shot at making a difference for all patients.
Right. I was just going to say this really does relate to the newborn screening. So, well, listen, I am looking at the time. It is 3 o'clock. I all very much appreciate the time you took today, and so excited to see what happens with this trial, and obviously we're going to be following it closely. So with that, I'm going to close the webinar, and it's my thanks to you again. So thanks a lot, everybody.
Well, we want to thank Parent Project for being so supportive, or we wouldn't be having this webinar. I mean, not the webinar per se, but the clinical trial per se, because we couldn't do this without funding support. And
Wow.
And Parent Project has put up the bulk of the funding to do the clinical trial.
Well, we're glad to do it. All right, everybody, have a good afternoon, and thanks again.